This is lecture 19, prokaryotes. Um, prokaryotes are the most ancient and most biochemically and ecologically diverse organisms on Earth. We'll talk a lot about that. We're giving a broad overview of their diversity, and this is part of structure and function, since we'll talk about a lot of adaptations that prokaryotes have, and they can be seen as adaptations for the huge diversity of environments in which we find them. The learning objectives are to be able to describe some important similarities and differences among all three domains of life, to understand and be able to describe the metabolic diversity of prokaryotes, understand how prokaryote photosynthesis and nitrogen metabolism works and why that is important for the evolution of all the other life on Earth, describe a few characteristics and examples of the major prokaryote groups, understand several functions of prokaryotes that are beneficial to other species and to ecosystems. So we introduce the three domains of life. Bacteria and archaea are the prokaryotes, and that goes along with a certain type of cell, a cell which lacks a nucleus. So prokaryote means before the nucleus, so you can think of that as what was here before eukaryotes, which are characterized by having a nucleus. That's what the name eukaryote means. So those are the three domains: bacteria, archaea, the prokaryotes, and eukarya or eukaryotes, the third domain. As we've talked about before, all of these three have glycolysis, the metabolism of glucose to pyruvate, releasing ATP, and this is evolved from a common ancestor. So the common ancestor to all three domains, to all cellular life on Earth, had glycolysis. They share a common DNA replication mechanism. Their enzymes are homologous in that the sequence of amino acids in the enzyme can be aligned with the inference that all of the DNA replication enzymes that all cellular life has evolved from a single set of enzymes in a common ancestor. And this is then true for transcription, translation, and the genetic code. They all have plasma membranes made of phospholipid bilayers, and they all have ribosomes for making proteins by translation. So we saw this tree before. This is the tree indicating the relationships. So we'll soon be talking about phylogenetic trees in detail. And this one we have ancient time in the left, present time in the right. We have three taxa: bacteria, archaea, and eukarya. And the tree depicts that archaea and eukarya in this group, in this set of three, are sister taxa. Eukarya are closer to archaea than they are to bacteria. Archaea are closer to eukaryote in terms of having a more recent common ancestor than to bacteria. Okay, we'll put some time marks on that in a minute. This table from the textbook gives the characteristics of the three domains and whether they're present or absent. So you can see that first line: the membrane-enclosed nucleus is present in eukaryotes but not in bacteria or archaea. It's absent in prokaryotes. Also, the membrane-bound organelles. Bacteria have a few, eukarya have many, and archaea generally don't have any. We'll talk about peptidoglycan. You don't need to memorize the other stuff, except to the extent that we talked about in the previous slide. Okay? But it is interesting to go through some of the similarities, some of the less well-known similarities. So for cell membranes, it's as we talked about with the evolution of cells, is one of the most fundamental structural aspects, the most fundamental structural aspect of a cell. Bacteria only have unbranched phospholipids in their membrane, whereas archaea contains some branched phospholipids. So those long chains, some of them have branching. And some archaea have a phospholipid monolayer membrane instead of a bilayer, so that the two phosphate ends, which are polar, are actually on the same molecule with a long chain, a long hydrophobic chain between. Only some archaea have that, and it's only known in archaea. So that's one variation on this phospholipid bilayer theme. So a couple differences, additional differences. So the first amino acid in proteins is generally formal methionine gets put in there. In archaea, it's methionine, and in eukaryotes, it's, as we've mentioned before, it's generally methionine. Still encoded by the AUG, but the translation system bacteria puts the formal methionine in instead of the regular methionine. So bacteria use a formal methionine transfer RNA. It's a distinct transfer RNA to do this, to bring the F-met formal methionine to the ribosome. Some other things that are interesting and useful biomedically. So there's different drug sensitivities of ribosomes. So certain antibiotic chemicals affect the function of ribosomes, and there are differences between bacteria and archaea in which ones are more effective than others. So for chloramphenicol and streptomycin, bacterial ribosomes are sensitive, but archaea ribosomes are not sensitive. Diphtheria toxin, which is from a bacteria, Corynebacterium diphtheriae, the ribosomes of bacteria are not sensitive, but the ribosomes of archaea are sensitive. So that's actually useful in the lab and clinically. So those are interesting differences. Why do those differences exist? We don't know. We don't have an adaptive explanation for them, although the assumption is that they could be adaptive. Remember that evolution works with whatever works at the time. So those cells evolved those changes. The lineage that gave rise to those groups evolved those changes because it was good for fitness at the time, or at least wasn't bad for fitness at the time. So these things are often called fixed accidents. It was what was available at the time. It became the structure or function. And then later events were contingent on that, or not. Or maybe it matters for other traits of the cell, maybe it doesn't. But we haven't really figured out functional explanations for why particularly those traits are present. So back to this table. So you can see that the archaea and the eukaryotes from our tree are more closely related. And to some extent they have some similarities, but remember archaea are still prokaryotes. They don't have a nucleus. So this slide shows the three basic cellular. There's a lot of structural diversity among bacteria. This slide shows the three basic cell shapes that have been characterized for a long time. And you hear biomedically in the classification of bacteria, especially those involved in infections. So we have bacilli, which are rod-like structures. We have cocci, which are more or less sphere-like structures. And we have spirella, which are spiral-like structures. So that's some of the basic structural divergence. But within those three, and there's other combinations, there's other types, there's a huge amount of diversity. Another way of classifying bacteria is through gram staining. And this is just something that was developed early on in microbiology to characterize how some bacteria are different from others. So this is a staining that involves a purple dye staining, and then you put iodine on the preparation. So this would be a sample on a microscope slide. And then you wash with alcohol, and then you add a red counterstain. Then you visualize under a microscope. If they strongly take up the purple dye, then they're gram positive. If they don't strongly take up the purple dye, then they're going to look more like the red counterstain. Those are gram negative. And it's been known for a long time that how this works is that the gram stain labels, it binds to peptidoglycan. And gram positive bacteria have a thick peptidoglycan layer. That's the brown in that diagram there, and you can see it in the cross-section electron micrograph there. Whereas the gram-negative ones don't bind much of the purple dye because they still have peptidoglycan, but it's a very thin layer. So you can see there's a fundamental difference between these two groups of bacteria and how thick their layer is, and that's just one example of diversity in the cell wall structures. And it turns out the bacterial cell wall components are pretty good targets for antibiotics, because if you disrupt the cell wall, you can disrupt the cell, and you can make the cell vulnerable to the outside environment. So, and you should also note that we'll hear again about cell walls in plants, algae, and fungi. Those are chemically very different cell walls. And as we'll talk about next lecture, the evolution of the eukaryotic cell involved the loss of the bacterial cell wall. And so those cell walls in plants, algae, and fungi evolved independently after that. So it's a distinct, chemically very different, structurally very different cell wall, but it's still called the cell wall. Go to question 17.1. So another very diverse feature of prokaryotes is how they move. So some species use flagella, and as we'll note later on, these are structurally different from the flagella that are present in eukaryotes. Flagella means whip. These are these whip-like structures, like in that purple cell to the right there. That's an E. coli cell, very well-known bacteria in our gut. It has flagella, flagelli. The singular could be flagella or flagellum. Other species have different ways to move. So this spiral-shaped spirochete has axial filaments that go up the length of the cell, so around that spiral, and their movement helps the cell have a spiral movement. Remember, these cells are all moving in water in an aqueous environment, so there's different ways of moving through the water. And a third way is to use gas vesicles and to control the gas pressure inside those vesicles to move up and down in the water column. So what you have there is a cross-section of a cyanobacterium, electron micrograph, and cyanobacteria is one of the groups that use these gas vesicles to move around in the water. There's also a great amount of diversity in how in the actual reproduction and exchange of genetic information. They don't have conventional sexual reproduction with recombination that we talked about earlier, but there's a lot of different ways in which they can exchange DNA. So generally, prokaryotic reproduction is asexual through binary fission. The genome duplicates, then the cell divides, and each cell gets a genome. And sometimes they might over-replicate, so a bacterial cell at any given time might have a lot of copies of its genome, such that when it divides, each of the daughter cells gets half of whatever those copies were. So that's called over-replication. That bacteria are known to do that. But there's no sort of programmed recombination like we know it from meiosis. Meiosis, remember, is a eukaryotic feature. Many species can undergo genetic recombination. There's well-known systems that involve plasmids. These were characterized early in microbial genetics in the middle part of the 20th century. So there's a well-known conjugation plasmid called the F plasmid in E. coli. And what you have there is an E. coli that has extended a pilus from its cell to another cell, and that's the ability to do that is encoded in genes in the F plasmid, and then through that little bridge there, copies of the F plasmid are going to be put into the cell that has the F plasmid, from the cell that has the F plasmid to the cell that was previously lacking the F plasmid, making it now have the F plasmid, so it would be an F positive cell. So F positive cells go around and convert F negative cells to F positive cells. We also know that bacteria can take up DNA in general from the environment, and sometimes instead of just eating that DNA as an organic food source, carbon source, it might actually use the sequences as functional genes. And we think that this is important in getting certain adaptations around between bacterial species, such as genes that encode resistance to antibiotics. Some of them are on plasmids. Some plasmids contain multiple genes for resistance for different antibiotics. It's a significant problem in fighting infectious bacteria, especially in hospitals. The main theme of prokaryotes really is about metabolic diversity. So remember, in our timeline, prokaryotes have had at least up to a couple billion years, or even more, than eukaryotes to adapt and evolve. They are found in every possible habitat on Earth, and they have much greater metabolic diversity than eukaryotes. So this is from the previous edition of the textbook. The current edition has a similar table. So we have 4 basic nutritional categories, and that classification is whether they make their own food through photosynthesis, or whether they use some other. So that involves light as the energy source, or whether they use something else for the energy source. So that would be the chemo part. The photo is light. The chemo is something other than light. An autotroph makes its own energy. Most of that's through photosynthesis, but some of that are through some other processes. and the carbon source comes from carbon dioxide in the atmosphere. So a photoautotroph gets its energy from light, its carbon from carbon dioxide in the atmosphere. That's a conventional photosynthetic organism that we'll talk about more, but it should be familiar. A photoheterotroph gets its energy from light, but it's going to get its carbon from other sources in the environment, organic compounds. So it's essentially made by other organisms, ultimately other autotrophs. So a heterotroph gets its carbon by eating other stuff. Okay, so there's some bacteria that are photoheterotrophs. Chemolithotrophs, which are also called chemoautotrophs, they actually get their energy not from light but from inorganic substances, certain molecules, certain ions. And they're lithotrophs or autotrophs, which means that they get their carbon from carbon dioxide, like photoautotrophs. So they have a way of fixing carbon. Chemoheterotrophs, so some bacteria and many archaea are chemolithotrophs, and those are some pretty odd environments where they're found, where there's not light, but they're these other inorganic substances that they can get energy from. And then finally, chemoheterotrophs, which are found in all three domains, get their energy from our organic compounds, get their carbon from organic compounds. So they're eating other organic compounds. So ultimately, autotrophs made their fixed carbon, and that's how they're getting that. And they use it for both energy and carbon. So which one are you? So most bacteria, archaea, all animals, of which you are an animal, fungi, and many protists are what? Do you get your energy? Do you get energy from light? No. Do you get carbon from carbon dioxide? No. So you are a chemoheterotroph. Okay. So we might ask you which one of these you are. You're a chemoheterotroph. So as we mentioned before, the evolution of photosynthesis took place in bacteria, and it was really fundamental to the evolution of all life on Earth and fundamental to evolution of the atmosphere and the entire planet. So without photosynthesis, planetary science of the Earth would be completely different. So photosynthetic bacteria are photoautotrophs. Light is the energy source. CO2 is the carbon source. So one of the most well-known types is cyanobacteria. They use a chlorophyll called chlorophyll A. They release oxygen as a byproduct, and they use water as an electron donor, and we'll hear more about the details of photosynthesis later on. This is like eukaryotic photosynthesis, and in fact, the eukaryote chloroplast is evolved from a cyanobacteria, and we'll talk about that next lecture when we talk about the evolution of eukaryotes. There are other photosynthetic bacteria which use bacterial chlorophyll. It's different in that it does not release oxygen. So some of them use, instead of water, they use hydrogen sulfide as an electron donor, and they release sulfur instead of oxygen. So it's kind of an analogous chemistry there. So even within photosynthetic bacteria, there's diversity in the chlorophyll that's used. There's diversity in the actual chemistry of the process. So some of these photosynthetic mechanisms are also used by photoheterotrophs for energy, but they're also able to get organic carbon. So they eat other organisms to get their carbon. So they might not get it from carbon dioxide. One of the important ones is purple non-sulfur bacteria. It's pretty well characterized. There's a picture of it right there. And in environments where there's a diversity of photosynthetic organisms, it's often been found that the peak of absorbance of the pigments, which are the chlorophylls, are spread out through the light spectrum, such that each group has a different frequency that they tend to use of light. So it's a way of dividing up this resource of light across the wavelength. So in that spectrum there, you see the purple sulfur bacteria. Their chlorophyll will have a peak way on the left end of the spectrum. And then some peaks of some other organisms, their chlorophylls might be like green algae, and some other photosynthetic organisms will have distinct peaks well spaced from those peaks. Because if you are up, say in the water column, you get the first light, you have your pick of all the spectrums. Your molecules are going to be tuned to whatever works best for you. So some part of that spectrum, you essentially absorb those wavelengths, and if whatever's below you doesn't have those wavelengths to absorb, they have to take what's left over. So you could think of them, it would be advantageous for them to evolve towards a different wavelength. And so by the time you get a few layers of photosynthetic organisms down through the water column, there might not be much left, but there might be some wavelengths that haven't been exploited yet. So then it would be advantageous for the photosynthetic organisms down below to tune their pigments to absorb whatever wavelengths are left. So what is chlorophyll? Here's a space-filling model of chlorophyll A. It's a big molecule. The dark grays are carbon, the light grays are hydrogen, the red is oxygen, the blue is nitrogen, the green in the middle there is magnesium ion. These work as molecular antennas to absorb light energy, and they use a complex process called resonance energy transfer, and a sort of chain of chlorophyll molecules which have sort of higher order structure where there's reaction centers in the chloroplast. This is at least in eukaryotes, but also in the cyanobacteria or photosynthetic bacteria cell. This will be a structure. These reaction centers transfer the electrons from the electron transport chain to the electron transport chain from these molecules, and the end of this process splits a water molecule to yield hydrogen ions and oxygen. So that's the ultimate. So these complexes of chlorophyll molecules that are organized into reaction centers, this is where this key process takes place. The chemical essence of photosynthesis is that 6 carbon dioxide molecules plus 6 water molecules plus light and chlorophyll in those reaction centers leads to a 6 carbon sugar plus 6 oxygen molecules. And we'll talk more about photosynthesis when we talk about plants. Here are cyanobacteria. They actually are primitively multicellular, and they have a form of differentiation among cells. So you have the spore there, the reproductive cells, you have the vegetative cells, and you have the heterocyst. The heterocyst is where nitrogen fixation takes place. We'll talk about nitrogen fixation in a minute. And to the right there is where you have a huge amount of growth of cyanobacteria in a freshwater pond because of eutrophication, which is where nutrients like phosphorus and nitrogen have gone into the pond, probably from agricultural runoff, and have really enriched the pond for growth of cyanobacteria, but that essentially shuts off the growth of anything else in that pond. It's called eutrophication. It's a big problem in both freshwater and marine habitats. And so these are two different species that do this, and in both cases they have some differentiation between nitrogen fixing cells and vegetative cells. But you can see everything's green, so the photosynthesis takes place in these different cell types, and these organisms grow colonially with differentiation among cell types. So because of their production of oxygen, photosynthetic prokaryotes made a respiratory lifestyle possible, the consumption of oxygen. So marine photosynthetic bacteria dominated the Earth for 1 to 2 billion years. Before this time, remember we talked about it, there was no oxygen in the atmosphere. So that there's massive production of oxygen once these groups evolved. They were very successful. They oxygenated the ocean. They oxidized most of the Earth, most of the iron on Earth, which is now mined as iron ore. And then after that, the percentage of oxygen increased to what we see today, and we had that chart of the constituents of the atmosphere from the last lecture. So respiration, which is how we get energy, is oxygen plus fixed carbon, glucose, leading to water, carbon dioxide, plus ATP. Now, we're all familiar with having to breathe oxygen and exhaling CO2. But that lifestyle is not possible unless there's oxygen in the atmosphere. And the oxygen was put in the atmosphere by photosynthetic prokaryotes and is continually put there by these prokaryotes as well as plants, which have chloroplasts, which evolved from cyanobacteria, as we'll talk about next lecture. Just an overview of aerobic respiration. You have oxygen coming in. This is in a mitochondria, which is, as we'll talk about next lecture, is actually evolved from a bacteria. So you have glucose first going to glycolysis, producing pyruvate, which eventually goes to lactate. And you have pyruvate going to acetyl-CoA into the Krebs cycle, producing more ATP, and then electron transport system producing even more ATP. And then out of the Krebs cycle, the CO2 comes out. So you won't have to memorize this. You would have to know this in detail in a biochemistry class, in physiology class. But just know that oxidative aerobic respiration is made possible because there's oxygen in the atmosphere that was put there by photosynthetic organisms. So another classification is chemolithotrophs. These are also called chemoautotrophs, as I mentioned. These use inorganic molecules instead of light as an energy source. So they might use ammonia or nitrite or nitrate or hydrogen sulfide, or sulfur. So whenever you have these things in significant quantities to be able to use them as energy, it's usually some kind of extreme environment. So one such environment, for example, where there's a lot of sulfur, is deep sea hydrothermal vents. So there's also, these are deep in the ocean, there's no light. So, but there's a lot of hydrogen sulfide coming out of the volcanic vents from deep within the earth. So there are prokaryote communities there which use this, they're chemolithotrophs that use this as their energy source. So another thing that prokaryotes do that makes all other life possible on Earth is what they do with the nitrogen cycle. So there's 3 steps of the nitrogen cycle. Prokaryotes perform all of these steps, and without any one of these steps we wouldn't have a nitrogen cycle and we wouldn't have available nitrogen. So remember, the atmosphere is 70% N2, molecular nitrogen. It gets there from other sources, and it's really kind of a chicken and egg thing, but originally you need to have nitrogen fixing, which is the next step. But on this slide we have denitrifiers, which put nitrogen into the atmosphere. So they're normally aerobic, but in some aerobic conditions you can take nitrate and an electron source and a proton source, and these produce nitrogen plus water. So Bacillus and Pseudomonas are 2 bacterial groups that do this, that are well known. So that's the green cells up top there are an example of one of those. It's a bacillus. Really the fundamental way that nitrogen gets taken from the atmosphere to be useful at all to any organisms is nitrogen fixation. So atmospheric nitrogen is converted by ammonia, for example, by bradyrhizobium, which is in that right picture there, those filamentous, those are root hairs which have nodules on them, so it's a soybean. So these grow in nodules inside root hairs of plants, where they're fixing nitrogen. It's down in the soil, but there's air there, and that air is 70% nitrogen. So in these specialized cells, these nodules, or those bacterial cells, performing this process, which needs to be needs to be actually completely away from oxygen, and it requires special enzymes. And because of this, we have fixed nitrogen, first in the form of ammonia, and then what happens then is nitrifiers convert ammonia or nitrite to nitrate. For example, nitrobacter in the soil in the ocean. You can actually buy nitrobacter for your aquarium. If you have too much ammonia, you can put nitrobacter in there and it'll help to get rid of your ammonia, convert it to nitrite and nitrate. And organisms can use either ammonia or nitrite or nitrate. But the bottom line, they can't use molecular nitrogen. Without prokaryotes, most other organisms would not have nitrogen to use. And bacteria, well, prokaryotes, are at each step of this 3-step pathway in nitrogen in the nitrogen cycle. So this slide depicts the nitrogen cycle. So we have nitrogen in the atmosphere, we have nitrogen fixation in plant roots, and the types of plants that have this are legumes. They're known for being able to do this in their roots. They're also known for centuries by agriculturalists because a lot of plants like corn take nitrogen out of the soil and leave it very nutrient poor. But if you then grow a legume like soybeans or beans or peas, because it does nitrogen fixation, it acts to put nitrogen back into the soil. So crop cycling is based on this. You want to do a legume and then a non-legume and back and forth. So that's been known for a long time. In the soil, there's also nitrogen fixing bacteria. So all of those are making ammonium. Ammonium also comes from detritus from decomposition, which takes place by a very diverse microbial community. Then you have, once you have ammonium, you have nitrifying bacteria, which are processing that ammonium for their own use, but they produce nitrogen in the form of nitrates. They transfer ammonium, transform it to nitrates and nitrites. And then you have denitrifying bacteria, which use nitrates or nitrites and put it back into nitrogen, atmospheric nitrogen, which goes back to the atmosphere. So denitrifying, you can think of that word as it's taking usable nitrogen away from the biotic environment and putting it back into the abiotic environment, the atmosphere, as a form that's only useful then to nitrogen fixing bacteria. So very critical, and it will pay to really memorize that process, the last two slides. So the vast majority of prokaryotic species have never been studied. There's some reasons for this. They're very small in size, generally they're microscopic. But more importantly, most cannot be cultured. The ones we know a lot about, we can grow very easily in the lab. But the vast majority of species cannot grow easily in the lab. We don't know their nutritional requirements. They might require to be grown as communities as opposed to single pure species. It's actually a lot of work once a new microbe is discovered, if you can have live cells, to actually try to get it to grow. It's a major accomplishment if you can characterize a new microbe and its growth conditions in the lab. It's actually a fairly rare development these days. But there's a modern field that uses DNA and DNA sequencing, when we've mentioned this before, called metagenomics or environmental genomics, where you take the DNA samples from the environment, from water, soil, or as we'll talk about, places in or on the body of another organism like humans, and you get the DNA and you sequence the DNA and you see what sequences are there. And you compare those sequences to the database. Some of them are going to be known, some of them are going to be related to stuff that's known, some of them might be completely unknown. And from that data, you can get a sense of the huge diversity of the microbial communities that must be present because their DNA is present. And that has led to a new science in the last 10 to 15 years, which is probably the fastest growing part of biology today. Very important for health, for medicine, also very important for environmental science, and has many applications as well.